Okay, set up. I'm recording now because then I'll post what we're talking about. And I'm going to actually be going over this briefly because we have a limited amount of time. One last piece of information that if we do wind up going remote for an extended period of time, big thing is about notes. Say like, if I were to say, if you're physically here and I had an opener up here, you would jot down that in your notebook, correct? But now when you're at home, how are you going to do it, especially if I don't see you for time and we don't want to deal with paper? Here's the easy fix. I'm going to ask you to open a Google Doc. We'll say honors biology notes, and then you'll start typing in and saving your responses as you go, okay? So that's the notes. Here, I'm going to go through this. Your homework tonight is I will post this video again where I'm going through this. I'm going through it rather quickly, these 12 slides in 12 minutes, so you're going to want to review the video and then jot down the most important things into your notes, okay? So again, the scientific method and controlled experiments. Again, science is a process of which we test, okay? We don't say things and you don't get to question it. We test things as best we can. So the steps, you have a problem. You want to research it. You're going to propose what is going to happen with that process. So you're actually making a prediction. You think it's going to work or not work. Then you perform the actual physical experiment. You collect data, which can come in two forms. Can have numbers, that would be quantitative, or it can be like in used words, qualitative. And then you draw a conclusion. And then there are more parts of this, right? You draw your conclusion. You have to publish, right? You have to get the information out there so people can critique it. That's the thing about what's going on in today's situation with the pandemic is people want this vaccine and they want it now. That's not how science works. To do these experiments and test and go through all the like these are usually decade-long type experiments. So again, the big push is we got to get this. Yeah, we know, but again, that's not how science works. Had you listened to the science in the beginning, we might not be as bad as we are now. And then you didn't listen, things exploded, and then they're like, oh, hurry up quick. We got to get healed. Well, again, that's not how it works. I once had a professor tell me, and I looked into it, and for the most part, it's true. If you look at any page in your textbook for any kind of science book, that's about 10 years for every page. Somebody studied and tested and researched those ideas for about 10 years. And again, over the time, the millennia, you wind up with your books. So again, but how do you test a hypothesis? Right? So you see the natural world. You ask in your head questions. You ponder it. Maybe you want to do an experiment. So how do you go about testing that hypothesis? So you're going to want, as best you can, you can't always do this, but you want to run what we call a controlled experiment. So that's where you're testing and isolating one variable. And then you have two groups. You have the control group, which all conditions are kept exactly the same. Then you would have an experimental group. So that's the group you're testing. You're going to keep everything the same except one thing. And again, you're going to want to go back. I'm kind of rushing because I'm taping. So again, just don't get frustrated because this is now your homework, right? You're going to go back, break it down, and get that information in. Let me ask you this. Would you be writing everything word for word? No. Put them in your own words and shorten it up because look what happens in this next page. Oh boy, that's a lot of information, right? So again, just try to listen to the words. So you've got your control group. The constants are those factors that remain the same. So you would put constant, means same, right? they remain the same throughout the experiment. You're going to compare the results of the experimental group against the control group to see if there are any differences in the results between the two groups. The test group has all conditions are kept the same except for one thing. You're changing one thing. The independent variable is the variable that's purposely changed in the experiment. So again, that one thing you changed, that is the independent variable. The dependent variable are the conditions that result from changing the independent variable. So you're going to test one thing, that's your independent variable, and then whatever happens, that is what your dependent variable is. It's the response. It's your response to the independent variable. So let's look at a hypothetical. A walk home from school. Suppose you want to figure out the fastest route home from school. You decide to try a different route each day for 1 week and time how long it takes to walk each route. So determine the constants, the independent variable, and the dependent variable. So what are some of the constants? What are some of the things that are going to stay the same? Your start point and your end point, right? So you're going to start from school and your end point is home. So those are constant. You're going to want to keep your pace the same, right? You don't want to run one day, jog another day, and walk. You want to, whatever your pace is, whatever your natural rhythmic pace is, you would want to keep that the same. Another thing you would want to potentially keep the same is, say, what you're carrying, right? You don't want to carry your book bag with all your books on Monday, because what is that going to do? It's going to weigh you down. It's going to affect your natural rhythm. So you want to be cognizant of that. If it's a warm day, like today and tomorrow, apparently supposed to be scorchers, right? Are you going to want to wear corduroys with a big hoodie? No, you're going to be sweating. That could, say, affect your performance. Say what you eat or drink, right? If you normally eat lunch and drink a certain amount of water, those are all constant. What do you think would be the independent variable? So the routes, so you're testing each route, right? Because you decide to do a different route each day. So yeah, the route is going to be. So what is going to be the response? So if you go home 5 different ways, what would be the response to those different routes? What would be different? Time, right? So in this case, the response would be the time to the independent variable, which is going to be the routes. Another one real quickly here. Let's look at what would happen. Suppose you would like to test the effect of a new fertilizer on plant growth. How are you going to make sure that the results of your experiment are valid? So let's break it down a little bit. Let's look at this one. So let's make some observations prior to the experiment. What are the 2 types of observations? So you got plant A, plant B. Your hypothesis, if fertilizer is added to the water, then what will happen? And then your experiment is to determine which factors need to be constant. So you got groups, you got water only, and then in the other group, the experimental group, that would be the control, you would have water and fertilizer. Yes, sir. Yeah. So you're going to run the experiment and you make an observation. You got A and you got B, and you can see something visually happen. So your data analysis. All right, so let's look at some data analysis here. So plant A, the height of the plant is 20 centimeters. Plant B, the height of the plant is 42 centimeters. conclusion, the use of the fertilizer does cause an increase in plant growth. In this case, the fertilizer was the independent variable, correct? Everything else was kept constant, right? Because you had water, you had a certain amount of water, but one group, the experimental group, had the fertilizer. What are some other things you would want to have kept constant? So yeah, so you want to make sure they both get the same amount of sunlight. What you didn't see beforehand, you just saw plants, right? So you would want to have made sure that the plants were in the same size pot, right? You would want to make sure the plants had the same dirt from the same source, right? So you didn't have any kind of outside contaminants from one source of dirt versus the other. So once you keep all those constants, then you're just adding water to the control, and then you're adding water with fertilizer to the test group, and you can see you wound up. So it's reasonable if you held that one, if you just tested that one thing, that it's a reasonable conclusion to state that fertilizer is the reason why the plant grew as much as it did. All right, so what you have here is an intro to scientific method. You going through this last, let's get to this last page. You are definitely in your notes going to want to note what these ones are, this very last slide, right? Because you're going to get an assignment tomorrow of which you have some experimental setups. And you're going to need to identify these parts. What was the control group? What was the experimental group? What were the constants? The independent and the dependent variable. So what your assignment today now is to go back through the video, hearing me, or even you can mute me because you want to get to the slides, correct? And then you get that information down. Remember, you're not doing things word for word. You're trying to get the big information. And these are important, these terms. So when you see these in some of the slides, you want to make sure you know what they are and understand what they are. But the big point of this is now expect some sort of recording along with the instructions in Google Classroom. It will not be me speaking 15 minutes every day. It might just be me conveying the instructions. So again,